we need to finish our discussion about cardiac output. See that I'm going to be all tongue-tied today. Okay, so we talked about how cardiac output can be affected by stroke volume, or the volume of blood that is ejected with every heartbeat, and the factors that can affect stroke volume. So basically, how much blood gets put in before it beats, and how much blood is thereafter, and how hard the heart is beating, and how much pressure the heart is working against to get that blood into those arteries. So now we're going to look a little bit at what affects heart rate, because that's the other side of this equation, because heart rate times stroke volume is, of course, cardiac output. There's a lot of different stressors that can affect heart rate, as I'm sure you've all experienced before, because certainly when we're under various stressors and definitely feeling various emotions, it can speed our heart up. Now, we talked about the pacemaker of the heart and the fact that the heart is setting its own pace and controlling its own beat, but the autonomic nervous system does affect heart rate. So we have hypothalamic areas and brainstem control centers that modify the pacemaker potential of the heart's intrinsic rhythm. So like many other parts of the body, the autonomic nervous system has dual innervation of the heart. So both divisions are going to have an influence, and the sympathetic nervous system is going to increase heart rate when it is active, both by direct neural synapsing using norepinephrine, as well as releasing them both as hormones circulating in the blood. The parasympathetic nervous system is going to decrease heart rate when it's in charge, when things are okay to relax. And as I'm pretty sure I have in another slide, sorry, I was checking, the parasympathetic nervous system most of the time actually maintains our heart rate at lower than it would be without any nervous system input. So the sinoatrial node, which typically sets the pace of the heart, as we talked about last time, it's going to be firing about 100 beats per minute, but resting heart rates for people are usually somewhere in like the 60 to 80 range. And that's because of the parasympathetic nervous system. So the primary control here is going to be in the medulla, in the brainstem. Literally, there's a region for increasing heart rate and a region for slowing it down. So cardioaccelerator and cardioinhibitory regions. And they're going to send their signals to something called the cardiac plexus, which is all of the nerves that are coming in to innervate the heart. The cardiac inhibitory is going to be the parasympathetic, and then the acceleratory is going to be this sympathetic. As you can see here, there's more innervation of the sympathetic side, and that's because the parasympathetic side really just affects the pace of the heart, whereas the parasympathetic also affects the strength of contraction. So the sympathetic regulation, whenever we are under some sort of stress where we think that we need more circulating blood, we're going to increase the signaling from that cardioaccelerator center, and that's going to lead to the heart beating not only faster but also stronger. So we're going to increase our cardiac output not only because we're increasing heart rate, but also because we're increasing contractility. So how much the heart is squeezing at a given stretch. Now for the parasympathetic, when it binds, it's not going to affect the strength of the heartbeat. It's just going to reduce the heart rate when we don't need that high sympathetic tone anymore. So we call this resting state vagal tone because the vagus nerve, remember cranial nerve number 10, is the one that carries these signals. And it's typically stronger, the parasympathetic signals are typically stronger than the sympathetic whenever we aren't stressed. So that's considered our resting state. It's what we should be at most of the time because it's not really healthy to be at a higher level, a higher heart rate all the time. So our resting heart rate is slower than if we had no nervous system input at all. And one of the consequences of that is that when people get heart transplants, their resting heart rate will increase because now the SA node is the one in charge all the time. And so their heart rate can still change because of other factors, but now there's no longer any nervous system control because you have to sever those nerves to transplant a heart, and we don't have a way to put them back together again, which is why I put this slide in. Even though it doesn't actually show it on there, trust me, they're being severed. So this is showing you, like, without any autonomic influence, the rate of the SA node. And then this is showing you how it slows it down with the parasympathetic regulation. So this is the same period of time. We have fewer depolarizations and therefore fewer beats. And then this is showing you that it gets faster with sympathetic stimulation. So just graphs showing you the same thing. Other things that can regulate heart rate, hormones can of course affect heart rate. So epinephrine released under the sympathetic nervous system control is going to have the same effect as the norepinephrine in the synapses. So that one just goes right along with it. Thyroid hormone will actually also increase heart rate and cause sustained increases. And that's for two reasons. One is that it has a direct effect on the heart because it's increasing your metabolic rate. If you remember that from endocrine last week. So with an increased metabolic rate, all of your tissues are going to have an increased need for oxygen and nutrients. So we need to send more blood through in order to fuel it. It also increases the cardiovascular system sensitivity to epinephrine and norepinephrine, allowing it to have a stronger response whenever the sympathetic nervous system kicks in to control it. The other big thing that can affect heart rate is ions. So the electrolyte levels in extracellular fluid, in the plasma of the blood, and the intracellular fluid have to be normal for normal heart function. If we have too much of an increase or a decrease in a lot of these ions, it's going to potentially have an effect on the electrical activity of the heart. Calcium is going to have the greatest impact, especially because increasing calcium levels in the blood and extracellular fluid are going to increase the contractility of the heart. But we also can see big impacts with potassium and, to a lesser extent, sodium chloride and possibly magnesium. You don't need to know that. I'm just babbling, I guess. And then obviously there are other things that can increase your heart rate or even decrease it, but drugs, okay? So stimulants, for example, like caffeine and nicotine are excitatory drugs in that they can increase heart rate, among other things. You don't need to know how it works, but if you're curious, caffeine will actually increase the frequency of depolarization at the SA node, and nicotine increases the firing of the sympathetic neurons because it's actually got a similar chemical structure to acetylcholine and binds to the receptors at the neurotransmitters. Other things that will potentially regulate heart rate, so things that will lead to changes in heart rate, exercise. So when you're exercising, your heart rate goes up. The more exercise you do over time and the more physically fit you are, the lower your resting heart rate tends to go because it's making your heart more efficient. so it doesn't have to work as hard to deliver the same amount of blood through your system. So in the immediate timing of exercise, your sympathetic nervous system increases your heart rate and your blood pressure, but in a long-term effect, we typically see them both lowered, both at rest and during subsequent exercise. Body temperature will have an effect as well. Basically, higher temperatures will increase heart rate, and that's because the metabolic rate of all tissues are increased with higher temperatures. So especially if you have a fever, you're going to see an increase in heart rate to supply all these tissues with more, with more nutrition. The opposite is true too, because hypothermia or an excess decrease in body temperature will actually slow your metabolic rate down. Your heart rate will slow down as well because it doesn't need to work as hard to deliver as many nutrients when your tissues aren't working as hard either. And then other factors, so age and gender. Obviously, there's not really a lot we can do about this, but generally speaking, our resting heart rate is high when we're born because we're so small. And then as we reach adulthood, it's typically lowering because we're getting older. And then as we start to actually age, we see it increasing again, the resting heart rate, because everything kind of declines after a certain point. And so we're going to become less efficient again. And see an increase in resting heart rate. And we also see a decrease in our maximum heart rate possible. In young adulthood, it's pretty high. We can get up to 220, but we see that decreasing with age so that we're unable to get our heart rate up as high as we get older. This is a nice chart summarizing all of that. Obviously, there's more information on here than you need to know. So just make your own version only with the information that you need to know on it, as well as here. But they're nice flows, and it kind of shows you a good way to set up the data to understand it. You don't need to know any cardiac embryology, but I think it's interesting just to see it because, you know, it's weird that we have this 4-chambered heart. How does it get that way? It turns out that it gets that way because originally we get these 2 tubes. So first we form these blood vessels, and these blood vessels have muscle in them that actually starts pumping on its own. And they fuse together, and then they do a little twist. And this little twist is how we end up forming our chambers. So basically it does this foldy thing, and we end up with a 4-chambered heart eventually, because then we still have to fully separate them. So that was kind of the last of the heart stuff. Did anybody have any questions about any of that? Again, you don't need to know the cardiac embryology. I just like to show everybody how weird hearts are. Okay, so we can always ask questions later if we need to. In the meantime, we will go on to the rest of the circulatory system. So the circulatory system, we break it down into 3 parts. We've got the heart, we've got blood vessels, and we've got blood. We're gonna run through the blood real quick, and then we'll end up on the blood vessels. Doesn't really matter what order you go in, honestly. I've never found an ideal order, but I think it's nice to just kind of hit the blood real quick. It's quicker, and then we can go on from there. So we're not quite going to do very much of these things. It looks like a lot, but we're just touching on a lot of this. We're going to start by looking at the anatomy of the blood and an overview of its functions. So blood is a connective tissue. Hopefully you remember that from our tissues chapter. And it's basically a liquid transportation medium. It moves throughout the rest of the circulatory system. So the blood vessels in the heart are a closed system, all connected, and the heart powers the movement of blood through. And we'll talk about how we achieve that blood flow in the next part. So connective tissues, you may recall, are characterized by having scattered cells and an extracellular matrix of a ground substance and protein fibers. So blood kind of has this. It has cells and some cell fragments, that's platelets, so we call them formed elements. And then the extracellular matrix is a watery ground substance called plasma, which is mostly water. And then the fibers are actually plasma proteins. So they are dissolved and suspended in the plasma. So they are not really fibers per se, but they're capable of getting there. And we'll talk about that in a little bit. We can divide up the functions of the blood into these three categories: transportation, regulation, and defense or protection. So the transportation functions of the blood are delivering needed things to the tissues of the body. So oxygen from the lungs and nutrients, either from the digestive system or from other parts of the body that are storing them. Delivering waste products for elimination. So carbon dioxide to the lungs, and then nitrogenous wastes, which are the result of amino acid being used. So amino acids make basically ammonia. So the blood carries these to their organs of elimination. And then the other thing that it transports is, of course, hormones. So lots of things circulating in that blood. Regulatory functions are basically the homeostatic mechanisms that blood is involved in. So it, of course, is involved in thermoregulation. And that's because blood can carry heat throughout the body. So like, you know, the warm blood from your core can warm up your extremities. If you're too hot, you can get rid of heat by sending more of it to the blood vessels under your skin, stuff like that. As we will eventually talk about, blood helps contribute to maintaining acid-base balance, so the normal pH of body tissues, but we'll tackle that a little bit later on in the course. And then there are things in the blood to help maintain a right blood volume. So maintaining adequate fluid levels in the circulatory system are important for maintaining blood pressure and for delivering nutrients, so transportation function. And so one of the reasons why we have proteins in our blood is to make sure that the right amount of blood stays in there all the time. The third function is protection or defense. So a lot of immune cells are circulating in the blood, as well as immune system related proteins and other chemicals that help protect our bodies from invasion. And then the system itself, or sorry, the blood itself is designed to make sure it doesn't get lost if blood vessels are damaged. So we have a whole blood clotting system, and we'll talk about that later as far as protecting ourselves from bleeding. So when we look at the components of blood, we find that the vast majority of blood is water and red blood cells. So red blood cells actually make up 45% of blood by volume, on average. There is a range. All of the other formed elements, so the white blood cells and the platelets, are just a very small percentage of blood. And then the plasma is 55% of the blood, and 90% of that is water. Within that water is protein, nutrients, respiratory gases, so oxygen and carbon dioxide, any hormones that are being transported, any waste products that are being transported, and then inorganic ions, especially sodium and chloride. There are three types of plasma proteins, albumin, globulins, and fibrinogen. We'll talk about in a second. And then the three types of formed elements, all of which we'll cover, are the red blood cells, so that 45%, which you can see over here. They're also called erythrocytes. And then white blood cells, which are also called leukocytes. And then these platelets, which are not full cells, which we'll address in a little bit, but cell fragments, which is why we call this formed elements and not cells. So again, you can see the red blood cells making up a large portion, the plasma making up most of the rest, and the white blood cells and platelets are just a tiny amount of that blood. This does give you some ranges for normal amounts of red blood cells. It does vary by gender. Women typically have fewer, so 37 to 47% of their blood is red blood cells, and men 42 to 52%. So the 45% is just an average. And then this is some pretty pictures of our formed elements, and we're going to work our way through them one at a time. But before we get there, we want to just tackle these origins of components. So the plasma proteins are mostly made in the liver. The liver makes a lot of things for the body. We'll talk about that more when we get to the digestive system. And then the formed elements are made in the red bone marrow, so inside of bones. So we have a lot of stem cells or progenitor cells in our bone marrow, and we are constantly undergoing this process of producing new blood cells and platelets. So that process is called hemopoiesis or hematopoiesis. This is for all formed elements of the blood. And then there are different kinds of stem cells to make other things or to make things. So basically our lymphocytes are made from lymphoid stem cells, and everybody else is made from these guys called myeloid stem cells because they're slightly different from each other, and we won't worry about it too much. So the different plasma proteins have different functions. All of them contribute to maintaining the volume of the blood, but albumin, which is the largest proportion or the largest amount of protein, largest proportion. Anyway, we have a lot of albumin. It helps transport various molecules that are lipophilic or hydrophobic, so ones that can't just be dissolved in the water of the plasma. For example, fatty acids and steroid hormones all hitch a ride on albumin. And then because it is so plentiful in the plasma, it helps hold the water in the blood. So osmotic pressure, or water's need to follow solutes, is how albumin, as well as ions and the other plasma proteins, are able to help. Because albumin is in larger amounts than the other plasma proteins, it takes credit for most of this osmotic pull. Globulins are the second category of plasma proteins. There's a couple of different types, and they have different jobs. So some of them are also transporters, like albumin. So they transport iron, they'll transport lipids, they'll transport fat-soluble vitamins. And then the third type is called gamma globulins. We're going to talk about them more when we get to the immune system. They're actually produced by immune cells, and they bind to invaders. So we'll cover that more when we get to the immune system, and hopefully that one will make more sense. And then the last type of plasma protein is called fibrinogen, and fibrinogen is actually part of our blood clotting machinery. We'll talk about how that works in a little bit, because fibrinogen can turn into fibrin, which is an insoluble protein fiber, and help clog up any damage to blood vessels to keep us from bleeding. So we'll cover how that works more in a little bit. This is just a little flow chart showing you how all of the different formed elements come about. You don't need to know any of this, but all of this is happening in the bone marrow, and then they all end up in circulation. That's kind of all you need to know. So we'll start with the erythrocytes, looking at a little more detail about how these different types of formed elements work and their functions. We're not really going to go through their life cycle, but we'll talk about the fact that none of these guys really live that long. So erythrocyte is the technical term. Erythro just means red and cyte means cell though, so it's just Latin for red blood cells, or RBCs. And red blood cells are pretty much the tiniest type of cell that we have in the body. One of the reasons for that is that they don't have a nucleus and they really don't have many organelles at all. So they, in the process of forming, they lose their nucleus, they lose their organelles, including mitochondria. And so because they don't have mitochondria, they can't perform cellular respiration. So in order to generate ATP, all they can do is glycolysis. And they're shaped like Lifesavers candies. So we call it a biconcave disc because it's got like a thick rim and then it's thin inside on both sides. So it looks like you could, you know, sit on it and raft down a river. And replacing all of these organelles is mostly just the protein hemoglobin. So hemoglobin carries oxygen. There are other things in there as well, but that's really the major thing. It's packed in there. And we'll talk more about that in a second. In order to maintain appropriate amounts of red blood cells in the blood, we have a hormone that stimulates that production, and it's called erythropoietin, or EPO. And I only mention it because it can be used by athletes to try to enhance performance. So if you've ever heard of blood doping, it's basically injecting yourself with EPO to make your body make more red blood cells, which is, sure. So this is what they look like when we look at them under a microscope. They're just kind of pale pink guys with paler centers because they are thinner in the middle. So their major function is, of course, to carry oxygen because of this hemoglobin. Hemoglobin is able to carry oxygen because there are irons inside of it. So it's actually 4 different proteins, each of which has an iron bound to a heme molecule. So heme is a pigment molecule, and every single iron is capable of binding 1 oxygen. And because there are 4 irons per hemoglobin, every hemoglobin molecule can bind 4 oxygens. Red blood cells typically have about 300 million hemoglobin. So that's, you know, like 1.2 billion oxygens per red blood cell. And there's a lot of red blood cells in our bodies. So we can carry a decent amount of oxygen. Hemoglobin kind of looks like this. It's like a 3D model. And when the iron binds to the oxygen, it kind of changes the shape, and that makes it look brighter red, which is how we end up with bright red arterial blood, which has a higher oxygen content, and a darker red venous blood because there's less oxygen in there. It's just a little shift. All right, so oxygen transportation is definitely the biggest function of red blood cells because of this ability to bind with hemoglobin and then be released from hemoglobin to deliver oxygen to tissues. It also does other things though, and we'll talk about this more when we get to the respiratory system. It actually carries carbon dioxide. So carbon dioxide is also able to bind to hemoglobin. Although it doesn't bind to the iron, it just binds to the amino acids of the protein part. And red blood cells do have other functions as well. They also help contribute to the acid-base balance of the body, which again, we'll talk about that more later. Stasis. So in a nutshell, they carry oxygen, and we have a lot of them. The next one up is going to be the leukocytes, and then we'll talk about the platelets. So the leukocytes are also called white blood cells, or WBCs. And again, just like erythro means red, leuko means white. So this literally translates to white cell. White blood cells are part of the immune system, so they are part of the defensive functions of the blood. Most of them do their job not in the blood itself, but out in the body tissues. So they do a pretty good job of protecting the body from all sorts of invasion, including sometimes the tumor cells that cause cancer. Our immune system is capable of killing some of those. So one of the hallmarks of white blood cells, though, because they work in body tissues, is the ability to exit the blood, which is a process called diapedesis, where they literally squeeze out of the blood, usually at the capillary. So this is just a single cell layer thick. They squeeze out in between the cells. And that's because when we have injuries from tissues, they give off chemicals that attract these guys. So we call that chemotaxis because they basically smell the damage and then they squiggle their way onward. So again, the blood is mostly just a highway for the white blood cells. They do most of their work out in the rest of the body. There are different kinds of leukocytes, and they have been named by differences in how they look under a microscope. So there's two categories based on whether or not we can see granules in their cytoplasm. So when we can see granules, we call them granulocytes. There are 3 of these. They all also happen to have a lobed nucleus, which means that instead of just having like this circle-y looking thing for a nucleus, it's all kind of like weird and blobby. All right. And then there are agranulocytes. There's 2 of those where we can't see granules. And those guys have like more of a normal looking nucleus, although it's actually a little bit bigger than in a lot of cells. So the 3 types of granular leukocytes are based on the colors that they look like when we put them on a slide and dye them so that we can see them. So we have neutrophils, eosinophils, and basophils. Here they all are in pictures. Eosinophil up top, neutrophil down here, basophil down here. And so all these little speckly things, those are the granules. You can see they're pretty chock full of them. It's harder to see on the neutrophils, but they are there. So the neutrophils are the most abundant leukocyte in the blood. Usually at least half of the white blood cells circulating are neutrophils. They do have this crazy long name too that you'll see. So they're also called polymorphonuclear leukocytes, or PMN. And that's because that lobe nucleus that they have, it has all sorts of different shapes to it. And this literally just means many shaped nucleus. So that's their other name. They have these cool granules in their cytoplasm that are really good at killing bacteria, because it has this thing called lysozyme that literally pops them, which is great. Don't worry about this part. Meant to delete it, missed it. So yeah, they kill stuff and then they eat it. So when cells consume pathogens, we call that phagocytosis. And they a lot of the times literally kill with something called a respiratory burst, so they attack them with oxygen molecules. Their job is to be the first ones on a site of infection or injury, so you can think about them as the first responders of the body. And then this is just a picture showing you a couple different ones so you can see how much variation there is with the nucleus. So this one's got a very blobby nucleus. And then this one's just like a horseshoe, and it can be anything in between those 2 things. Eosinophils are fairly uncommon, only 2 to 4% of white blood cells. They typically have 2 to 3 lobes to their nucleus, and their granules are really pink. Eosinophils are thought to have originally been like the parasitic worm or like, yeah, internal parasite branch of our immune system. So they attack the kinds of parasitic worms that can invade our bodies that are too large to be phagocytized by neutrophils. And so the reason why they're uncommon in the blood is because most of them are living in loose connective tissues, especially around our intestinal tract and our skin and our lungs. We see their numbers increase in people with allergies and asthma. We also see them increase in parasite infections as well as certain autoimmune diseases. And that's because there's definitely a connection between all of those things. So this is what they look like. I think they're pretty, but this is sort of showing you the bilobed, and this one's kind of more of a trilobe nucleus. And that leads us to the third granulocyte, which is basophils. Basophils are very rare. They're less than 1% of the total number of white blood cells in the blood are typically basophils. They are also a bilobe nucleus, and they're covered with very dark granules. And these guys, again, we don't see them a lot because they're in tissues, and they're actually quite pro-inflammatory. So they will increase inflammatory responses for things, especially when it comes to parasites and allergies. And usually what happens is then the eosinophils come in afterwards and try to clean up their mess. So they look like this. It's just a lot of dark spots. Hard to tell where the nucleus is compared to the granules. And for comparison, this guy's another neutrophil, or you can see these nice light staining ones. See if she stops. All right, so that's 3. We have 2 more, and these guys are the agranular leukocytes or agranulocytes. So now these guys don't have visible granules, and we have 2 kinds, lymphocytes and monocytes. So monocytes are the 3rd most common leukocyte, and they're still less than 10% of the total. They're probably the largest ones, and they have this kind of horseshoe-shaped nucleus, or like partial moon-shaped nucleus. These guys also do most of their work outside in body tissues. So their job is to go out in the tissues, become macrophages, and macrophages also phagocytose pathogens and damage tissue. So these guys come after the neutrophils and finish cleaning up the mess. So again, more pictures of their weird little nucleus. And then this is what it looks like when they go into the tissues and become macrophages. So that's one active, angry macrophage trying to do its job. The last type are lymphocytes. Lymphocytes are mostly B cells and T cells. So there are different types, not C, Jesus, T. There are different types of lymphocytes, but in the blood, we can't tell which one's which. Now we'll cover the different types of lymphocytes when we get to the immune system, because like half the immune system is basically these guys. Now they're the 2nd most common leukocyte, so 20 to 30% circulating in the blood. And the different types have different functions, but all of them are basically immune related. So we'll hit the details of those functions in the immune system. For now, we just need to know that they're, 2nd most common, and they circulate. And they just have like, it's pretty much just all nucleus with a little bit of cytoplasm. Nucleus, a little bit of cytoplasm. That's what these guys look like. So the difference here is that if it was a monocyte, it would have that little indent. All right, so 5 types of white blood cells, and that's all the cells in the blood, but we have one more formed element, which is the platelets. So platelets, again, they are cell fragments. So there's no nucleus here. It's just a bit of cytoplasm with a bunch of granules with a lot of different chemicals in it for a variety of functions. And overall, platelets help stop blood loss. So when blood vessels are damaged and you're bleeding, one of the things that helps that stop happening is these platelets. So this is kind of just showing how they get derived. There are these giant cells called megakaryocytes in the bone marrow, and they just kind of stick themselves out and slowly break off into platelets. So you can see here's a red blood cell. Red blood cells, like I said, are quite small for cells, and these are the little platelets. So they're much smaller than a cell, so they're cell fragments. So hemostasis is the general thing that these platelets are involved in. So we're just going to go through hemostasis, and you'll see how the platelets play a role. Now, there's actually a couple of different steps to this. So when we say hemostasis, we just mean stopping blood loss. And then the fancy term for blood loss is hemorrhage. So there's a couple of different things that make this happen. We have clotting factors, which are various proteins and stuff in the blood. We have platelets, and then we have the lining of the blood vessels themselves, which are endothelial cells. So that's that simple squamous epithelium. Hemostasis is a rapid, local, and controlled response to damage and bleeding. And the reason why is because if it happened excessively, we would actually have problems. So we need to prevent excessive clotting because if we're sending blood clots into the circulatory system, we're going to potentially be blocking perfectly good circulation. And that's how we can get things like aneurysms and not aneurysms, strokes, and like losing parts of your lungs because you throw a clot, heart attacks, stuff like that. So we divide up hemostasis into 3 stages. All of them are triggered by damage to the blood vessel. So vascular spasm is the first thing that happens. Basically, damage to the blood vessel causes the smooth muscle in it to constrict in response to injury. So we're literally decreasing the diameter of the lumen, which makes for less room for the blood to escape. And this will give us about 30 minutes of decreased blood loss, which isn't that bad. And the idea is that it's giving the other mechanisms time to kick in. The second thing that happens is that the platelets go into action. So they form a plug. They literally plug the hole in the blood vessel wall. And the hole is much smaller because of the vascular spasm shrinking it down. So whenever we have exposure of platelets to collagen fibers, which is what happens when you have exposed blood vessel wall, they start getting sticky. They stick to the collagen, and then they release their granules with chemicals in it, and then they stick to each other. This process is called platelet activation. They also get larger, so they swell, and they all kind of just stick together, and they also stick whatever else is coming through to them, like red blood cells and other proteins and stuff like that. And so they form this nice plug to allow time for this third part of the process to kick in, because the third part is going to last for as long as needed to fix the tear. This guy is good, but it's a temporary fix. All right. So just another diagram. So the platelet plug gets us started, but coagulation and the formation of blood clots are actually going to seal the blood vessel tear so that it can heal back up again. Coagulation involves clotting factors, which are also called procoagulants. And when we start forming blood clots, it's a chain reaction that leads to the formation of fibrin. So 12 different substances are clotting factors. Most of them are actually plasma proteins made in the liver and circulating in the blood as zymogens. Zymogens are inactive enzyme precursors because we don't want this to happen unless it actually needs to happen. Now, 4 of them actually require the presence of vitamin K in the liver for synthesis. And that's important because that means if we don't have vitamin K or if something blocks vitamin K's activity, we will stop making these and we will eventually start spontaneously bleeding. And this is how some of the rat poisons work. And that's scary because sometimes kids or pets will eat them. And if we don't know about it, we might find out because they just start bleeding. One of the substances is something called tissue factor, which is actually present on the cells of the deeper layers of the blood vessels. And another one is actually calcium. So calcium has a bunch of important roles. Not only is it important for things like neurotransmitter release and, cardiac muscle function, it also is necessary for coagulation. So what happens is we start this chain reaction and we start activating these zymogens, and each one activates another. It's like a row of dominoes or an avalanche or something like that. And what happens at the end is that fibrinogen that's been circulating, it's the last zymogen to activate. And when it activates, it becomes fibrin, and fibrin falls out of solution and forms this meshwork patch that works with the platelet plug to form a blood clot. So it's a classic example of a positive feedback loop. No, it's one of the few positive feedback loops we have, right? So once we get this started, it happens over and over again until the stimulus is gone. And that stimulus is exposed blood vessel wall. So now all you need to take from this is that the formation of these things leads to the formation of more of it. Whereas we've talked about negative feedback loops before, this is our positive feedback loop. And all of it results in fibrin formation. The last step for making a good, strong blood clot is retraction. So with clot retraction, the platelets, because remember they got all poofy and sticky, they all contract. They actually have proteins that let them all pull together, and they're attached to the fibrin, and so the whole thing gets squeezed and shrunken, and this squeezes the serum out, which is the remaining liquid portion of the blood without the fibrin in it. So plasma is all of the liquid of the blood, and then serum is liquid portion of the blood with the fibrin out of it. So question, what causes nosebleeding in some people? Usually that's caused by things that damage the blood vessels in the nasal cavity. Most often it's going to be dry weather, related things that dry out the inside of your nose, although there can be other things as well. So although it's possible for somebody to get nosebleeds because they have a bleeding disorder, it's more commonly other things that just make the blood vessels get damaged. And then they stop eventually because you're still able to do this. So yeah, the clot retracts, and this helps bring the blood vessel edges closer together so that there's a smaller hole to patch up. And then the platelets actually have growth factors that they release that stimulate the blood vessels to heal. And so then the idea is you heal it up and then you don't need the clot anymore, and then you dismantle it. So that's blood clots and hemostasis. Now there are definitely a lot of different ways that hemostasis can go wrong. Some of them are inherited issues, and some of them are caused by diseases, and some of them are caused by toxins. So the rodenticide that causes bleeding because it blocks vitamin K can certainly be a problem. There are some diseases that cause problems. So there's a disease where our immune system attacks our platelets, for example. Diseases affecting the liver, since the liver makes most of the clotting factors, will also be bad. And then there are definitely various diseases of platelets, diseases where people, or not diseases, but disorders, where people don't make all their clotting factors, or where their blood vessels aren't quite right, where they're going to be born with an inability to have normal hemostasis. Yeah, so there's a lot of ways it can go wrong, and how we treat it depends on what's actually going wrong, because we have to treat the right thing. And I do believe that this is just like an overview of hemostasis, or disorders of hemostasis. It can be very interesting if you're curious, but you certainly don't have to access it if you don't want to. The last thing we like to talk about with blood is blood types, because we all have one and you might need to know what it is at some point. So we're just going to go through what blood types are, what the options are, and how we figure out who has what to know who can get what kind of blood. And then we're not going to go over that because like I said, trying to cut things out for time. So blood types are basically caused by proteins on the surface of red blood cells. They're called surface antigens, and we're not really sure why we have them, but we know that they're there and that there are consequences. And that's because there's different types of these surface antigens, and not everybody has the same ones. So in order to understand what a surface antigen is, we need to define an antigen. Antigens are anything that the body can generate an immune response to. And the most common immune response that we see is the formation of antibodies, which are proteins that will stick to the antigens so that they can no longer have an effect. So basically they are meant to stick to things that are coming off of bacteria and viruses and toxins and clump them together so that they can't cause harm. That clumping is called agglutination when it happens to red blood cells. So this is what an antibody looks like. It's a couple of different proteins stuck together in these little Y shapes, and on either end of the Y, on the two short ends, they can bind to one specific type of antigen. Antigens all have different shapes, and each antibody can only bind to one particular shape of antigen. We'll talk a little bit more about this when we get to the immune system, but hopefully this is enough for blood typing for you guys. So we actually make a bunch of different types of red blood cell antigens. In fact, we have identified at least 30 groups of antigens, and that means there's more than 30 antigens. It means there's 30 groups of them. Only 2 of those 30 groups cause significant transfusion reactions, so negative effects of receiving blood from somebody else. So we only worry about those in most people. So we're going to talk about each one. There's the ABO blood group and the RH blood group. Now, transfusion reactions are what happen when somebody gets blood from the wrong blood type. So when blood types are incompatible. And basically what happens is if somebody has antibodies against another person's blood type antigens, they will have an immune response to this blood that they're receiving, and then they'll just, like glom onto the red blood cells. They will clump in your blood vessels, and then they'll get destroyed. So you're having this like, ooh, sorry, I think I just got a delivery. Oh, scared me. Because that was totally my doorbell too. Give me 1 second, sorry. Okay, so yeah, so that's going to cause some bad things to happen. You're going to have like some inflammation, you're not going to feel well, and you might have some bad effects of clumps of stuff getting stuck in small blood vessels. So we try to avoid that. And in order to do that, we need to be able to know what blood type you are and what type of blood you can receive. So the ABO blood group is based on 2 antigens. So there's A antigen and B antigen. And there's 3 options for what you can have on your own blood cells. So you can have both of those antigens, and in which case your blood type would be AB. You can have either A or B individually, but you can also have neither. And when you don't have either A or B, then we say your blood type is O. So as our immune system develops, we typically make antibodies against whichever antigens are not present in our own body. So our body knows not to have an immune response against whatever's in our own blood cells, but it also automatically has an immune response against whatever we don't have. And so we call them anti-A antibodies and anti-B antibodies. Let's not worry about that. Taking that out. All right. So the way this works. So here is somebody with A antigens on their red blood cells. So their immune system knows that A antigens are good because they're part of our own self, but they don't have any B antigens, so they automatically make antibodies against B. So anti-B antibodies. So because they have these antibodies, they cannot receive any blood with type B antigens on it because they would have a transfusion reaction. So they can receive blood with A antibodies because that doesn't cause a problem, and they can receive O because O has nothing on it for these antibodies to bind to. If we look at people with type B blood, we see it's the opposite. So they have the B antigens, and so their immune system knows not to have a problem with the B antigens, but then they end up making the anti-A antibodies because their immune system doesn't know any better. So that means that they can't receive any blood with A antigens because then they would have an immune response. So B isn't going to cause a problem because that's the same as their own blood, and O isn't going to cause a problem because, again, there are no antigens to bind to, there are antibodies to. Now, people who have both A and B, they don't make antibodies because now both are part of their own body. So there's no antibody production. And that means that they can receive all types of blood because all of them are compatible. They have no antibodies to have a reaction to. So type A blood and type B blood are safe. They can receive type AB because, I mean, that's their own blood type. And then O is also safe because there's no antigens to bind to. We see the opposite case with type O blood. So they have neither of these antigens on their red blood cells. So their immune system is going to make the antibodies to both. So they have anti-A antibodies and they have anti-B antibodies. And that means that if they receive blood with either A or B, they will have an immune response. So the only type of blood that they can receive is O, just like they are, because there's no antigens for their antibodies to attack. So because O can only get O blood, they are kind of screwed. But we like having them because they can give their blood to everybody, which is why we call them universal donors. And people with type AB blood make horrible blood donors because they can only give their blood to other people with type AB blood. But if they need blood, they're in luck because they can take blood from anybody, which is why we call them universal recipients. Anybody have questions about this? Okay. Now, the RH blood group is a little bit more straightforward because although there are a bunch of different versions of the RH antigen, almost everybody just has one type if they have it. So they either have it or they don't. And so we just need to worry about whether they have it or they don't. So we just call it RH factor. It's called RH because rhesus monkeys are the first species that this was recognized in. So that's where RH comes from. So if they have it, we say they're positive, and if they don't have it, we say they're negative. Now, positive is far more common. So we know that 85% of Americans are RH positive. The proportions of blood types do vary by country, though. So there are differences there. Now, another difference between the RH group and the ABO group is that our bodies don't automatically make antibodies. So we don't have any antibody formation unless we're exposed. So basically, if you're Rh negative, then you don't have antibodies against Rh unless you receive a transfusion of Rh positive blood or are otherwise exposed to it, and then your immune system makes antibodies. So we call that process sensitization, where the exposure to the antigen is necessary to trigger the immune response. So that's a weird difference between the Rh blood group and the ABO blood group. When we report people's blood types, we typically report them together. So you get a letter for which antigens you have from your ABO blood group, and you get a positive if you have Rh factor, and you get a negative if you don't. So for example, an AB positive person has A antigens and B antigens from ABO and has Rh factor from that group as well, which also gets called D. O negative means no antigens on their red blood cells from any of these things. So these are the extremes, and everybody else falls somewhere in between. So when we test for this, we actually expose your blood to antigens, got that wrong, to antibodies, and we see what happens. So basically, your red blood cells are going to have their antigens on there, and we send in the antibodies, and if you have the antigen, then they bind and you clump. So when it clumps, it looks like this one and this one. If you send in an antibody that you don't have the antigens for, then nothing binds and nothing clumps. And so then you just have a little spot of blood hanging out with some fluid, and that's what that looks like. So this person, for example, we know that they have A antigen because we got clumps, which means that the antibody's bound. We know that they have Rh factor because we got clumps here too, so that means that they had Rh factor for the antibodies to bind to. And we know that they don't have the B antigens because there's no clumping, so that means that there were no antigens for the antibodies to bind to. So this person is A positive. So they make these handy little flow charts for the rules for transfusion to make it easier to follow. It kind of just goes over the same stuff that I talked to you about, so you can definitely review based on this. Now, things get complicated when you try to add them both together. So, you know, that's fun, but you don't really have to remember that. If you're having trouble understanding how this works, there's a cool website with a game on it that you can play to help you study this. So let me show you. Where did you go? There we go. Okay, so the Nobel Prize is, of course, an international award that's given for various reasons, and a lot of them are science related. And they have a website where they make various informational things related to some of the things Nobel Prizes have been won. So there is a blood typing game, which apparently requires you to run Flash. It's nice. It's going to go well. That stops working. Oh boy, that's fun. It's awful. And so there's some nice stuff about this. So first of all, it does give you some information, and it also lets you play bigger games and shorter games. So let me show you this. Oh, and if you go to the main menu, it has tutorials. So it's a nice way to get more information about what I've already told you what you need to know, but so you pick a patient, and first you have to sample some blood. So you actually have to like maneuver the syringe over their arm. And then you have to put some blood into your 3 test tubes, which all contain the antibodies for these various factors. So we put all that in and we mix it up. And if we click on this, it shows us that there are antibodies in here. All of the antibodies. We don't seem to have any antigens though, because there's been no clumping. So if there's no antigens, then they have type O blood because there's no A or B, and there's no antigens for RH, so they're negative. So they're O negative, which sucks for them because then we don't have a lot of blood to give them. And then it takes you over and you're like, okay, but this guy still needs blood. So which blood can they receive? Now, I wouldn't recommend just doing this until you get a handle on it. Luckily, he only needs 1. Now, O negative people can only get O negative blood. So if you give him anything else, it's going to have a transfusion reaction. But here's the O negative, and you drop it there, and then he's magically fine. So I really enjoy this game. I think it's the best one for helping people understand blood types. So if you're having trouble with that, you can just play this game, and it'll help. So when a person that's Rh positive has a child that's Rh negative, basically, and it is in the textbook if you want to read about it. So it's called hemolytic disease of newborns. And I did cut it out because we don't have time to cover everything. But basically, there is risk to babies that are born, but the risk is actually the other way around. So when a mother is Rh negative, if she has children that are Rh positive, then there's a risk that her antibodies will attack the baby. And that can potentially cause miscarriage. At the very least, it's going to cause anemia when the baby's born. And there is treatment for it. So if you want more information, just start with what's in the textbook. And you can also just look up hemolytic disease of newborns and find some information about that. I usually cover it, but because we don't have as much time as I do in my 2-semester courses, I cut it out. And this is, again, if you look up the blood typing game, it'll take you to this. There are other ones, and there are some interesting infographics and stuff. If you guys have kids that are of the right age range, or just if you are interested in some of the science stuff, I highly recommend going to their website and checking out what they have. All right, let's go back to the slideshow. Okay, so this one I just put up there, not because you need to know it, but because I like to show how complicated it gets. Here's some various resources about blood types, including some of the rare blood types that we didn't actually talk about. This is mostly about the rarest blood type, and actually it includes some information about hemolytic disease of newborns, so check that out if you find blood typing interesting. And that actually takes us to the last part of the circulatory system, which is blood vessels. And of course, we're like almost out of time. So that sounds about right. So let me just get this started, and I guess I'll have to finish it up later. I can do a bonus lecture on Friday for you guys to watch over the weekend. So blood vessels, we want to start with anatomy, and then we have a couple of physiology things to talk about. So there's 3 different types of blood vessels, and we covered this a little bit already. So arteries carry blood away from the heart, and from the heart, as they go, they actually branch smaller and smaller as they eventually deliver stuff to tissues. And veins carry blood towards the heart, and they merge larger and larger or converge and join until they reach the heart. So in between veins and arteries is capillaries. Capillaries are the smallest blood vessels, and they exchange materials with the tissues. And so we have capillary beds in between these branching arteries, and then they converge back together to these big veins. So it kind of looks like this. So arteries leaving the heart, they branch until they become capillary beds, which then converge back together into larger and larger blood veins, which then return blood to the heart. Most blood vessels have a 3-layered wall surrounding the lumen, which is the space inside with blood. And we call those layers tunics. So just like, I don't know if we've covered something with that before, but just like some other things. Now there are exceptions, and those exceptions are mostly the capillaries and other teeny tiny blood vessels. But all the larger blood vessels have this setup. So the innermost layer is called the tunica intima, and it is the endothelium. So this is continuous with the endocardium. It's all the same layer of simple squamous with a basement membrane and a little bit of supportive connective tissue. The middle layer is called the tunica media, and if it has muscle, which a lot of them have smooth muscle, it's located in here. And then there's also frequently elastic fibers so that it's able to have some stretch and give. The outermost layer is the tunica externa, and this is a little bit of connective tissue, so mostly collagen fibers that protect blood vessels and also anchor them, so attach them to surrounding tissues. So how thick these layers are, just depends on how big the blood vessel is. The larger the blood vessel, the thicker these layers. So this is showing you the 3 layers in an artery, and this is showing the 3 layers in a vein. Typically, arteries have thicker musculature, so thicker tunica media, and often they have more elastic, and that's just because they usually work at a higher blood pressure. Something else that this is showing is the blood vessels within the walls of the blood vessels. So just like the heart has blood vessels, so do the larger blood vessels because they're too thick to receive nutrition from the blood in the lumen. We have 3 different types of arteries, and they all have different jobs. And we're going from the largest arteries, so coming right off of the heart, like the aorta, and we're gonna branch down to smaller. So the largest ones are elastic arteries. Because they're really large, they have really thick walls, and they have the largest amount of elastic too. Their job is to expand and recoil as the heart ejects blood. And the reason why they do this is because they have to act as pressure reservoirs. So when they receive an ejection from the heart, they're gonna bulge a little bit as they take that extra blood volume, and then they will retract back. And what that does is it maintains smooth blood flow even though the heart is pulsing out blood. So you're getting this volume and then nothing, and this volume and then nothing. And these guys are going, okay, we got more and then let it back out again. And we get more and let it back out, okay? So they help maintain a steadier blood pressure and flow of blood. As we branch into smaller blood vessels, we get to these distributing vessels. These are the muscular arteries because they proportionally have more smooth muscle and less elastic. Their job isn't to bulge now. Their job is to change their diameter to control how much blood is flowing to specific body organs. So then as we branch into these body organs, we get into the smallest arteries. So the smallest arteries are called resistance vessels, and they are arterioles. So they're so small that they don't count as arteries anymore. They are arterioles. Now we start to lose that 3-layer tunic. So sometimes we have like literally a single cell layer of smooth muscle around an endothelium and like barely any connective tissue externally. These guys determine which capillary beds get which blood flow. And because they are so small, even small changes in their diameter will change how much blood is flowing through them, which we'll talk about that in a little bit. Or probably I'll talk about it on Friday. So this is comparing them all. Obviously, this is differences in scale. These guys are microscopic, and so they're much, much, much smaller than these guys, even though they're showing them all like they're the same size. Once we get past arterioles, that's when we enter these capillaries. So capillaries are the smallest blood vessels. They're literally just a single layer of endothelial cells. And most tissues are just chock full of them. They just have a ton of capillaries, and the capillaries are these like interbranching, interconnected webs. So we call them capillary beds. And the presence of capillary beds provides tissues with perfusion. So, so a blood supply for nutrition and stuff. There's actually 3 types of capillaries, and they're based on their relative permeability or leakiness. The most common type, the type that we see everywhere, is the continuous capillary. So continuous capillaries are the least leaky, so they're not super permeable compared to the other ones. They are pretty good at controlling the flow of materials through their walls. So they have a lot of tight junctions in between the cells, which prevents larger things from squeezing in between. There are small spaces called intercellular clefts that allow water and small solutes dissolved in the water to squeeze in between, but most stuff has to move through the endothelial cells to get out. So most of the body is continuous capillaries. There are fenestrated capillaries in places that need to filter more stuff out. So fenestrations are holes or windows or pores in the cells themselves, like they literally have holes in them, to increase the leakiness. Now there is a basement membrane, so a diaphragm of glycoproteins covering these guys still. So that does control some amount of what's able to leak out. We see this in places where there's active filtration, absorption, or hormone secretion, where we need to move more stuff through. So for example, we see this in the kidneys, and we'll talk about that when we get to the urinary system. The last type is called sinusoid, and sinusoid capillaries are barely even there. So not only do we have fenestrations, but we have these gigantic intercellular clefts, and we have holes in the basement membrane itself. So giant stuff can go through. So cells can actually fit through. So we only have this in places where we want cells to be able to move out of the blood. So that's in the liver, the bone marrow, the spleen, and the adrenal medulla. Those are the only locations where we have capillaries that are this leaky. Now, capillary beds aren't just there. Capillary beds are all interconnected in these networks, and they are controlled by a single arteriole controlling blood flow in, and a venule, which is the smallest type of vein, controlling blood flow back out again. This setup of an arteriole to a bed of capillaries to a venule is called the microcirculation, because all of this is, of course, completely microscopic. There's two types of vessels in this capillary bed. So there's one that's not actually a capillary, it's actually an arteriole. It's called a vascular shunt or meta-arteriole. And its job is to allow blood to bypass the capillary bed. And then there's true capillaries, and the number per shunt depends. So somewhere between a 10 and 100, which you guys don't need to know that. It looks like this. So basically we have an arteriole, and then we have the arterial. So this is controlling the blood flow here. We have smooth muscle cells wrapped around here. And then this meta-arteriole has some smooth muscle cells at the base of every single capillary. And if blood is going to flow through all of this, then these guys are going to be relaxed. If we want blood to bypass this capillary bed because it doesn't need that much blood right now, then all of those squeeze shut as sphincters, and the blood just goes straight through, bypassing the capillaries and out the venule in what we call the thoroughfare channel. So how that's determined is based on various factors, and we'll address them a little bit more later. But the job is to supply any tissue bed that needs blood with blood, but to not give extra to anybody that doesn't need it, because we only have a certain amount of blood to go around, and not everybody gets all of it all of the time. So we'll talk about that later as far as like perfusion regulation. But mostly it's about local chemical environment and nerve fibers controlling those smooth muscles. So mostly the autonomic nervous system. So that's going to take us to the veins. And so the venules are the smallest veins. So as the capillaries come back together, we get venules. The very first venules are actually part of the perfusion exchange, just like the capillaries. And then as they get larger, they start adding on smooth muscle, and they're no longer able to perfuse. and then they come together into veins, and veins, just like arteries, have the 3 distinct tunics, the same ones. The difference is that they always have less smooth muscle. So the tunica media is going to be thinner. The lumen is going to be larger. And then the tunica externa is thicker. So we have they're more of a thin, floppy blood vessel. And that's because their job is to act as capacitance vessels and blood reservoirs. So their job is to hold more blood if we don't need our full blood volume circulating. So up to 65% of our blood volume can be held in our veins when we're at rest, and then they can kind of squeeze with what smooth muscle they have and put it into the rest of the system. Capacitance is their ability to kind of just expand and hold stuff. Another difference between arteries and veins is that some veins have valves. So just like the heart, where we prevent backflow with valves, these veins have it too. They're typically like a 2-flap thing, and they work just like the semilunar valves, where the flow of blood backwards closes the valve cusps and prevents blood from going back. We don't see them everywhere, but in veins that are always flowing against gravity, where there's more likely to be more backflow, like in your legs, we see a lot more veins. There is another type of vein, and those are the sinuses. So we kind of addressed these when we talked about like the central nervous system. Veins that are sinuses are flat, and they have very thin walls. So they rely on surrounding tissues for support. And so these are just kind of in places where traditional veins don't work, and we need kind of larger but better shaped or more ideally shaped things, just like these venous sinuses in the meninges. All right, so since I've managed to use up all of our time, I will do a catch-up lecture Friday afternoon. I'll post the specifics. You're all welcome to join me, but don't feel obligated to. And I will post it on Friday evening so you can watch it over the weekend before we talk about the respiratory system on Monday. You have a question? The video is not up on Mondays. I will make sure it goes up. Let me look. Hold on. Go away. So oh yeah, I apparently forgot to post it. All right, so it'll go up, right? Is that what I did? That's crazy. What was I thinking? I don't know. It's been a weird week. So yeah, it'll go up. I'll make another page on the links to recorded lectures because it recorded, and I'm pretty sure I posted it. I guess I just didn't actually link the post. So sorry about that, but I will fix that right now. Any other questions before we call it a night? All right, per usual, email me, and I am working on getting everything graded. Whenever they're graded, they'll go up on Canvas. You can always see that. And otherwise, I will see you all on Monday, and I will be getting a circulatory system worksheet or practice quiz, if you want to call it that, up this weekend as well. All right.